So it's already 12. I think I'm going to start my introduction. So to make sure we have plenty of time for discussion at the end. So welcome everybody to the Connect Science seminar series at Dana Farber and Harvard Cancer Center. Please type your questions at the end of, or whenever during the talk in the Q&A option, not in the chat, and I will read them at the end. Today, it's my pleasure and a great honor to introduce to all of you Dr. Nir HaKohen. He is the director of the Center for Cancer Immunology at MGH. He's the David P. Ryan Professor at Harvard Medical School, and he's also the director of the Center for Cell Circuits at the Broad Institute of MIT and Harvard. Dr. HaKohen obtained his bachelor's from Harvard University. Then he moved to California for his PhD in Stanford University, and then he came back to the East Coast for his postdoc in immunogenetics at the Whitehead Institute at MIT, and has since then stayed here as a PI. Dr. HaKohen is a true pioneer in the use of system-level approaches, combining computational and really deep, thoughtful mechanistic immunology. He's done amazing work in, from describing genetic variants that explain different control of pathogen sensing in dendritic cells, to identifying different disease subsets in humans with single cell work. I actually have to say that he came to Mount Sinai when I was a postdoc. I think it was in 2017 to give a talk. And that was one of the best talks. And that talk opened my eyes to the power of single cell technologies to deepen our understanding in immune function and immune cell types, with work that I think was published, I would say 2017 in Science, describing the different disease subsets. I really enjoyed that talk a lot. And he's done also lots of work in immunotherapy, in melanoma, in colorectal cancer, and today he's going to talk about immune hubs. He already published work in immune hubs, but I believe this time is spatially resolved immune hubs. So I'm really looking forward to this talk. Thank you so much for accepting our invitation.
Yeah, thank you, Judith. And that was a great visit to Mount Sinai. I really enjoyed that as well. And so I'm glad that was fun for you at that time. All right, so this is great to be here, and this has been an amazing series. So I'm very happy and honored to be speaking. So what I'll talk about today is, as Judith said, is spatially organized immune hubs in tumors, and in a way, kind of using systems approaches to learn the rules of human cancer immunity. And there are many factors that can drive tumor immunity, from the antigens themselves to the different immune cell types and their interactions, to evolution of the tumor and the immune response, and so forth. What I'll talk about today is really the immune cell types that are present in a tumor and their interactions that contribute to tumor immunity. So we'll have three parts of the talk. The first one, I'll review a paper we published last year, inferring and localizing hubs of immune cells and malignant cells in colon cancer. I'll tell you about unpublished data we have on identifying and refining these hubs further in lung cancer and predicting outcomes of checkpoint immunotherapy. And then finally, targeted therapy to manipulate the chemokines that organize these hubs. And this work was led by Karen Pelka and Jonathan Chen, Matan Humphrey for the colon cancer, and led by John Chen for lung cancer, and targeted therapy together with Jun Tian and Ryan Corcoran at MGH. So the initial puzzle we wondered about is there spatial organization to immune responses in tumors? Lymphoid organs are very organized, but we don't necessarily think of tumors as having physically organized, spatially organized immune responses. The second puzzle to us was, why is it that MMRD or what is different about the immune responses in mismatch repair deficient tumors versus mismatch repair proficient, which at this point is very well known in the field, particularly with the neoadjuvant trials recently showing in some cases 100% response rate and cures in MMRD tumors. So this is incredibly effective immune response to anti-PD-1 therapy in mismatch repair patients, which unfortunately are the minority, and almost none in the mismatch repair proficient tumors. So we wanted to understand a little bit about the differences in a productive immune response versus a non-productive immune response. And so we initially, in the first study I'll talk about today, looked at 62 patients with colorectal cancer, about half mismatch repair deficient and half mismatch repair proficient, and collected at MGH both tumor and adjacent normal, and together with DFCI and Brigham, brought this collection to be a very nicely balanced collection between the two types of colon cancer tumors that I was just mentioning. And we wanted to know what are the cells and gene programs that distinguish between these two types of programs, focusing, of course, on the immune cells and the immune aspects of other cell types. And we wondered, given the data that we generated, could we infer multicellular interaction networks? And then ask, how do the cells that we hypothesize are in these networks, how do they participate in interactions in spatial hubs? So that's what I'll talk about in the first part of the talk, and then we'll move to lung cancer and targeted therapy. So summarizing it quickly, we identified across these 60 or so patients, 7 lineages of cells. Of course, we know, you know what they are: epithelial, stromal, and then the different immune cell sublineages, generating about 87 clusters of cells. And what we found was that CXCL13 positive T cells, specifically both CD8 and CD4, were highly enriched in MMRD compared to MMRP tumors. You can see, for example, here, the CD8 compartment, CXCL13 positive cells are highly enriched in MMRD tumors, but much lower levels in MMRP tumors, and almost none in the healthy adjacent or the adjacent healthy tissue. And here you can see another view of it, which is showing, for example, in the CD8 T cells, the clusters that I was just showing you, for example, number 14, you can see these have a high cytotoxicity score. Of course, it has the expression of the CXCL13 associated program. It also has high MHC and interferon gamma response genes expressed in them. So these cells, we believe, are both cytolytic as well as expressing exhausted markers, and I'll come back to what this means in a moment. But then we were able to look independently at bulk TCGA, and you can see that the CXCL13 program is higher in both our samples, of course, but in TCGA, in the MMRD versus the MMRP. So we believe these are probably the key differences in T cells. There are others as well, but I won't discuss those today. What's interesting about these CXCL13 positive T cells is when we stain for CXCL13 to find these T cells, we see that they're spread throughout the tumor. And in fact, not so much in the tertiary lymphoid structures, which do express also CXCL13 to attract B cells, but that signal is not primarily in the T cells. So they're spread throughout, and then based on Giacomo Oliveira's work and several other groups over the last couple of years, there's a finding that these cells that express CXCL13 are the tumor reactive T cells. This is basically shown by cloning the TCRs, the Giacomo had done in Kathy Wu's lab, the DFCI, and identified these cells as the ones that are tumor reactive, both neoantigen and non-neoantigen reactive. So the CXCL13 is a good marker of these. So we believe that these CXCL13 positive T cells that we see enriched in MMRD are basically showing us the tumor reactive T cells that are spread out throughout the tumor. We then looked at other cell types, macrophages and monocytes. These are increased dramatically in both MMRP and MRD. To look more carefully at the subsets that are enriched, so we have monocytes and macrophages. However, these don't cluster particularly well in subtypes. What we found instead was that looking at programs of gene expression, so that is co-regulated gene expression programs that may actually be present in multiple cells. For example, if you look at an interferon stimulated gene program, program 21, you can see that it's present in multiple areas of this monocyte macrophage lineage. So we believe that looking at these programs is a better way than asking to find subclusters of these monocytes and macrophages, because some programs will actually be turned on in different subtypes of cells. And that's important because that is what's critical during a dialogue between cells. Certain gene programs turn on. It's not that the one cluster necessarily is the key responder, but that programs turn on across clusters. And so, so I think the program approach is particularly important for malignant cells. Malignant cells, as people who have looked at single cell data know, separate usually by the different sources of tumors. Different patient tumors on the UMAP will be separated. So a way to address that problem is, again, to look for gene programs other than clusters. And here are the different gene programs we identified in malignant cells in these samples. So you can see that there are, for example, cell cycle programs and many other stem cell programs and other metabolic programs. But we focused in, again, honed in on the immune programs. There were 3 major programs, one an inflammatory one and 2 ISG programs. And you can see here the different patients and their level of usage of that program in the malignant cells. You can see the MMRD and MMRP. So in general, the MMRD will tend to have higher programs for immunity. So you can see here the MMRD has higher expression of this program, this inflammatory program, as well as these interferon stimulated MHC2 programs, which are likely stimulated by the type 1 and type 3, type 2 interferons. So, so these are higher in MMRD and MMRP, both in our data as well as in TCGA. So, so now we have 3 kinds of programs I've shown, 3 kinds of differences: the T cells that are tumor reactive, the monocytes and macrophages that have inflammatory programs, and the malignant cells. And so I didn't have a chance to show you today because we'll focus on other things. We also found that fibroblasts also develop very unique programs that have to do with controlling the stem cell niche and maintaining it, as well as inflammation in the context of wound healing. But these are the ones I want you to remember and focus on right now. So the next question was, given these programs that exist that are more in MMRD and MMRP, can we infer multicellular interaction networks for these? And so the hypothesis would be that if 2 cells were interacting with each other, then the programs of gene expression that they express, that they have in the cell, would correlate with each other if you looked across tumor specimens, if the cells were communicating with each other. So, so for example, if you had these programs here, P2 and P3, those gene expression programs, those co-regulated gene programs, would go up together as you go from one sample to the other, or inversely related. But in any case, some relationships that suggest that they're talking to each other, or that some other signal is coordinately turning both on, but suggesting in any case a coordinated response between these cells. And this is not something we would find easily by looking at clusters, because again, clusters don't represent necessarily the communication pathway. Sometimes they do, but the programs are a more general way to find them because they both find clusters as well as sub-programs that could be across clusters. So when we look at the correlation structure of these programs across different cell types in MMRD, MMRP, you can see we find hubs of enriched correlation. So you can see here there's a very large hub here that we'll see in a moment, we'll call the immune hub, and that's not present in the MMRP tumors. But you can see there are different hubs in each one, and each of these can be studied to understand the potential interactions between the different cell types. Hubs, this hub 6, which we're going to focus on, is the hub that's interesting because it actually contains the programs I showed you earlier, which we associated with MMRD, but not MMRP. And so the T cell programs that I mentioned to you, the CXCL13 program, as well as interferon gamma MHC2 program and the T cell proliferation program all together, also correlated with the malignant cell program that expresses these MHC and ISG programs, as well as with myeloid programs that express ISGs, as well as with dendritic cells. So these programs are correlated to each other across patients, and you can see the individual programs, again, are higher in MMRD patients, shown here, and then in MMRP, and of course not really present in adjacent normal tissue. And if you look at the correlation structure with these graphs, what you find is that these programs are highly correlated to each other across samples in MMRD, but that correlation structure is very weak or almost lost in the MMRP tumors. So it really is a connectivity and a communication that occurs in the mismatch repair deficient tumors. And when we look closely at some of the genes that are expressed in this system, what we find is that the CXCL13 positive T cells, many of them express interferon gamma, which is exactly the stimulator of the programs that I mentioned in the malignant cells, the macrophages and the dendritic cells, and vice versa, that the chemokines that are produced in these three cell types are actually the attractors of the T cells, and these are CXCR3 positive T cells that are present in the tumor. So this provides essentially a positive feedback loop where the incoming tumor reactive T cells are stimulating the factors that bring in more T cells and create a positive feedback loop. So what does it look like spatially? So the first thing we did was to use the nanostring geomics technology, which allows you to take regions of interest throughout the tumor, and we took many, many regions of interest. And there we were able to stratify cells into either pan-CK positive or negative, which tells you whether they're malignant cells or not malignant cells, and look at the expression of, at the RNA level, of many genes in these different regions. And what you find is that the ISG score in the malignant cells is correlated to the CXCL13 positive cells present in these regions of interest. So not only across tumors is there a correlation between the different programs, but also within a tumor, you can see the different regions, there's a correlation, further supporting this kind of positive feedback loop and spatial localization between these two programs. And so, but one can look even at higher spatial resolution and stain for these chemokines, CXCL1011, that bring in these T cells, as well as CXCL13 and interferon gamma and CD3 to mark the T cells. And you can see that they form these hubs that are spatially localized throughout the tumor and represent these localizations of these cells together. And one can then dissect a particular hub and look more closely and find both the expression of the chemokines in the pan-CK positive cells, in the pan-CK negative cells, and the presence of T cells in this area. And so this is the data that supports that, and we can do a more unbiased analysis of the entire tumor, of all the tumors, and look for these clusters in an automated fashion and find that these hubs that we find, in fact, contain expression of these programs. This is an example here of the T cell CXCL13 program, and the epithelial cell tumor ISG program is present in these hubs and is higher in MMRD and MMRP patients using just the staining. So, so what we conclude as a model here is that the T cells are producing the factors that then create the conditions by which these malignant dendritic cells and macrophage are able to then produce factors that further attract the T cells. And interestingly, a paper came out at a similar time in mouse model from Deborah Fowl, together with Andy Lusters and Joanna Groom, finding that these types of hubs also exist in the mouse and actually in a vaccination model with CXCL10 positive niches together with T cells. And these were perivascular here. So we believe this is probably the same types of niches or hubs, as we call them, that we find in the tumor. So they may be very generally existing. And so to conclude this part of the talk, we predict immune hubs based on the covariation of transcriptional programs across tumor specimens. We do a spatial analysis to find that, in fact, even within a tumor, these are correlated to each other. And then doing very deeper staining, we can identify the hub as these foci of malignant stroma and immune interface. And we believe these are really the tumor reactive T cells that are present based on our other findings, that this is the best marker actually across all tumors for tumor reactive T cells. So the next question is, can this tumor be found in other, can these hubs be found in other tumors? Can we predict can you predict response to immunotherapy? Are there subtypes of tumor immunity hubs? So I'll address these different questions. We find that the tumor immunity hub is in fact generalizable across tumor types. We see them in lung and breast. And so we believe that they are there. And this was work done by John Chen, a pathologist in the lab, together with Max Burrell and our clinical collaborators. So we believe that these are really everywhere. And so we then decided to do a deeper study together with Justin Gaynor in non-small cell lung cancer. And indeed, we use the same markers again, these chemokines and the T cells markers, and can identify these same spatial hubs that are present in non-small cell lung cancer. And so, but we want to look a little bit deeper this time and look at the T cell subtypes within this hub. And so we looked at the different exhausted T cell subtypes. There are these progenitor cells, there are some activated cells, and then there's terminally exhausted cells. And so we want to use markers for T cells to look at that. And so we created a panel that would allow us to look at progenitor exhausted T cells that are usually present in tumors, as well as the activated and exhausted T cells that we see in tumors. And so using these markers together with Ki67 for proliferation, we were able to stain tumor sections and then basically co-index them with the other panel that I showed you earlier, where we looked at the CXCL13 positive T cells and the CXCL10-11 attracting centers. And so we're able to enumerate hubs throughout the tumor and identify these immunity hubs based on expression of CXCL10 and 11. This showed us that about 1% of the tumor actually contains, of these many tumors that we looked at across all samples, contain immunity hubs. Now, using all the markers that we had just stained with, we were able to take these different regions with immunity hubs and subdivide them based on the markers into different clusters. So this is very similar to what you would do in single cell analysis, but here instead it's in each spatial hub is essentially pulled into different clusters depending on the markers that are present. And what we found was that clusters 5 and 7 in particular show up as associating with the responding patients. So these are now patients who have received anti-PD-1 therapy, and we separate them into responders and non-responders in non-small cell lung cancer. And you can see cluster 5 and 7 are highly enriched in responders. So you can see that very much here. And what's interesting is that what we found is when either subcluster 5 or 7 are not present in the tumor, then this person really is going to be a non-responder to anti-PD-1 therapy, while if either subcluster 5 or 7 is present or both, this increases the probability of response dramatically. So what are clusters 5 and 7? What subset of the immune hub that I showed you are they? And when we look at the different markers to see which markers were enriched in clusters, subclusters 5 and 7, which are present in different parts of the tumor, we found that their subcluster 5 in particular is enriched in the CD8 positive TCF7 positive cells. This is an interesting subset because these are the progenitor cells that are essentially present in some tumors at high numbers, but in others not. And we had found in earlier studies that the presence of these progenitors can be indicative of a good future immune response. Now, these progenitors are the ones that generate the exhausted T cells and the effector T cells that actually kill the tumor. But without these progenitors, several mouse studies had shown for many groups, including locally here, Anna Anderson and Nick Haning's group and others, this is a key progenitor for required for the anti-PD-1 response. So we found that the cluster of the subcluster 5 of immune hubs that contains both the CXCL10-11 as well as the TCF7 positive T cells is an important predictor of immune response to anti-PD-1 of clinical response, as is the cluster 7, which is has some TCF7 cells, but mostly has the interferon gamma positive T cells that are probably the more effector-like T cells and exhausted T cells that are doing the killing. So the combination of cluster 5 and 7, we believe, is probably an important, particularly important clusters to have to predict response. Now, in the previous study I showed you in colon cancer, we didn't know who would respond to immunotherapy or not, although by taking MMRD versus MMRP, we obviously enriched one versus the other. And so here we actually know which patients are responding and non-responding. So this really confirms that these spatial hubs are important for the ability to respond. And these are all shown pre-therapy. And so these TCF7 positive hubs are very unique and are found pre-therapy. And they also contain markers of dendritic cells such as CCL19 and CCL22, but really not so much B cells. So B cells we think of as being in tertiary lymphoid structures. We find them very rarely or in small numbers in these hubs. And we found that if we do a biased analysis to look for all structures that contain TCF7 across the tumor, we find that subcluster 5 that I told you about earlier really overlaps perfectly with these TCF7 areas. So we believe that cluster 5 and 7 really are the key clusters that you need to have to produce an effective immune response against PD-1. And the hypothesis is that these hubs form these hub 7, for example, which is probably the hub of the cells that are actually killing actively, and then the CCL19 hubs that I just mentioned that contain the TCF7 as well, which we have been calling hybrid hubs because they contain both CXCL10 as well as the TCF7 positive T cells and CCL19 dendritic cells. They're kind of similar to what we see as tertiary lymphoid structures, which are often found outside the tumor, but again, they don't contain the B cells, so they're not complete TLSs. And when we use geomics again from nanostring to look at expression levels of genes in the hubs that are inside these TCF7 hubs versus of immune cells that are able to provide a niche for T cells to be essentially activated from the progenitor state. And so dendritic cells are present here, the T cells that are the progenitors, and that the hypothesis is that these will then be activated, become proliferating cells, and enter the immunity hub that really contains the malignant cells and the myeloid cells that express CXCL9, 10, 11, and the ISGs, and then can basically form a a structure that allows really effective killing of the tumor. So essentially what you're doing is creating a organized region for tumor destruction and an organized region that protects progenitor T cells from the tumor so they don't become overactivated and go entirely to exhausted. And the reason you have dendritic cells in these TCF7 structures is to enable the presentation of antigen from the tumor locally, get the activation of the T cells, and then move them into these hubs, which then presumably go and kill and destroy different parts of the tumor. Now, interestingly, Joanna Groom had described in lymph nodes that these two kinds of structure may exist there as well, with the TCF7 or TCF1, which is the same gene, forming here in the T cell paracortex. But in the interfollicular zone, you find that you get the activated T cells along with the chemokines that bring in CXCR3 positive T cells. And so we believe this kind of structure that we see, these two structures that we found here in an unbiased way, really reflect very nicely the two kinds of structures that protect and activate the TCF7 positive progenitor cells and have activated T cells in another structure. So we believe this organization exists as well. And the tumor probably evolutionarily is very similar. So in the ongoing work that we're doing now, we're using higher multiplex approaches such as MERFISH to identify these hubs in an unbiased fashion. And there we are able to identify these regions of CXCL9, 10, and 11, and identify that CCL19, for example, which is the marker of dendritic cells, is present there, as are markers of macrophages C1q and CD163, suggesting really that these hubs contain the cells that we initially had inferred from correlation structure. And also based on these findings, we're John Chan, again, as a pathologist, so interested in finding out how this can be used clinically, has developed a two-color in situ hybridization that could be used in the clinic that would basically identify these hubs through the existence of CXCL10-11, which forms when these immune hubs form, and CCL19, which again is a marker of these TCF7 hubs that are allowing the progenitors to form. So we believe the combination of these two markers can be predictive, and that is one of the projects we're working on now. So to answer some of the questions I was asking is, do these immune hubs exist in other tumor types? Yes, they do, in multiple tumor types. Are they composed of, do they have different subtypes? When we look more closely at the T cell states, looking from the progenitor to the effector and the exhausted and proliferating, we find that indeed these hubs contain the different subhubs or substates of the hubs contain different T cell states. And that these particularly two subtypes of hubs, the TCF7 containing T cell hub and the interferon gamma T cell hub, are really enriched greatly in responders versus non-responders, and can even predict the response to therapy. So we believe that the tumor, the spatial organization in the tumor mimics that of lymph nodes, with a region that protects the stem-like T cells, enabling a positive feedback loop between the T cells, the malignant cells, and it protects the stem-like, and a second one that enables the feedback loop between the T cells, the malignant cells, and the myeloid cells. So you get both an area where the T cells can form, and that basically maximizes your ability to expand them, and an area where they can do their work and kill the tumor. And these need to be separated because if the progenitor cells were just mixed in and not protected, they would become hyperactivated, and we would lose those progenitors. They would differentiate too quickly toward the exhausted phenotype. All right, so can we enhance immunity hubs? And this is work doing with Jun Tian and Ryan Corcoran at MGH, who have been very interested in the question of MAP kinase inhibition and targeted therapy for several years, and finding that there is a MMRD tumors are actually more sensitive to MAP kinase inhibition, and that you can get some really good responses in patients who are MMRD to MAP kinase inhibition, which is a key in colon cancer for BRAF mutant tumors. And so when Ryan had looked at some of the basically bulk RNA-seq of paired biopsies from patients who had been on MAP kinase inhibition, and there are different combinations, in this case BRAF, MEK, and EGF receptor inhibitors, you can see that the responders had actually higher T-cell signatures, and that these increased in time from day 0 to day 15 biopsies using the bulk RNA-seq. And similarly in mouse models, there was also an increase in T-cell infiltration with the combination of the BRAF MEK inhibitor, and of course even more with BRAF MEK and PD-1. And what June had seen is that there's a cooperativity when you add the BRAF MEK inhibitors with the PD-1, you get a better control of tumor in mouse models that contain BRAF mutant tumors. And so, so I think the key point here is that these observations were important to test in a human system. And so Ryan Corcoran developed a clinical trial around BRAF V600 colorectal patients with therapeutics against BRAF and MEK, as well as anti-PD-1. And so many of the patients were MSS, and so the idea and the hope was that one could stimulate some of the responses that we see in this synergy with MSS patients. And in fact, there was a pretty good overall results for this, with a nice waterfall plot that you can see here with this combination. And so we wanted to look at these patients and understand what were the changes that occur. And we identified that patients who had progression-free survival less than 6 months versus greater than 6 months had a change from pre to on that was different. So here you can see, for example, the T cells had a greater increase in patients that had longer survival than those who had progression-free survival that also had lower progression-free survival, suggesting that the immune response was important. And when we looked specifically at the epithelial cells, the malignant epithelial cells, we found that in those that survived that had PFS greater than 6 months, you could see these ISG signatures very similar to the ones that we had seen in our studies so far, in what I've shown you, the programs that were turned on. But those with less than 6 months did not have such a strong ISG response. So you can see it here again, engine response genes in response to interferon gamma and alpha. Chemokine activity was higher in those with PFS greater than 6 months than those with less. And interestingly, this was tumor intrinsic. We were able to look at the malignant cells again directly and see this higher epithelial ISG score. If we took the program that we found from our previous studies of colon cancer, same thing here. These ISG scores were higher in on than pre, and greater in PFS greater than 6 months. And the MAP kinase score, which is a score to tell you how well your MAP kinase inhibition works, of course, also went down and was more effective in patients with a PFS greater than 6 months. And so we looked to see now in organoids, which had been generated for many of the patients, whether there was any correlation there. And what we found was, in general, there was a bit of a greater inhibition in organoids of a greater inhibition of the MAP kinase signature. You can see here below, this is the MAP kinase signature going down, and this is the ISG signature going up. In general, when you gave stronger blockers of this pathway, BRAF/ERK inhibitor in particular, you could see you had both higher signature of the ISGs and also lower signature of the MAP kinase. And so these were inversely proportional here. And what's interesting there is that this is no immune cells are here. This is just the organoids, just the malignant epithelial cells. So essentially what you're seeing is that there's an intrinsic induction of this program, the one that we had found correlated with the immune hubs in these patients. And so what we concluded from this kind of study is that in addition to seeing potentially an improved patient response in this trial, we found that there was greater MAP kinase inhibition in patients who had better outcome and greater induction of the same immune programs that we had seen go with responders in both lung and in the MMRD versus MMRP. And amazingly, that the patient organoids also suggest that there really is a tumor intrinsic innate immune response driven by these MAP kinase inhibitors in inducing those responses. So we believe that this is we are now looking at the spatial structure to see, to try to understand a little bit more about that. But the idea is that perhaps the MAP kinase inhibition in malignant cells triggers an intrinsic program that then would attract the T cells and would further enhance the spatial hubs that I told you about earlier. Okay, so that's what I wanted to end on, because I think that's an interesting potential direction to go in terms of enhancing hubs. There may be other ways as well. And I just want to thank the team that did the colon work that we heard about earlier, primarily Karen, John Latan, and Cici Sarkazova, who worked on developing this together with Aviv Regeb and many others. And then the lung team, which was led by John, Jen, who I mentioned before, also working with Linda Neiman, Justin Gaynor, and Max Farrell. And then finally, working with Brian and his lab, Brian Corcoran's lab, and Jun Tian on the MAP kinase approach. So, overall, what we're imagining is that understanding these hubs ultimately will allow us to better control them and to better enhance them in different situations, or perhaps reduce them in the case of autoimmunity if we find them there. So that's kind of the overall theme, and I'm happy to take questions.
That was great. Thank you. So please type your questions in the, okay, they are already. Bart Letterbach, I apologize because I'm surely mispronouncing your name. Doesn't forced CXCL10 expression increase T cell activity in vitro?
Right, that's a great question. We have not tried enforcing CXCL10 expression. I would guess that that some people have done that. I think that it would certainly increase the attraction of the T cells to the organoids. If we did that, that would not be surprising, because the activated T cells are going to be expressing CXCR3, and so they would certainly be further engaged and brought in, and that would in turn then activate them further, because they would come closer to the tumor and form this positive feedback loop. So, we think that this is certainly a way in which, if one could enforce it, it could be a really interesting way to further increase these hubs. So yeah, I think it's a good point.
So Keith Loeb is asking, how does the function of the intertumor hubs differ from TLS function?
Yeah, so TLS, people have not really proven exactly what the TLS do in the tumor per se. There's certainly been observations of them, and they've been associated with outcome. A big question with the TLS is what the B cells are doing. What role are they playing? How important are they for tumor immunity? These are questions that still haven't been answered. I think the primary thing that we see here is that these hubs are present closer to the tumor. The ones we found are present closer to the tumor. And again, there are two kinds. There's the ones that are really the activated T cells with the malignant cells and the macrophages, which we think are the active hubs. The TLSs are certainly not those. The TLSs are far away from the action. And we believe those are a way for the tumor, for like evolutionarily, if you have a virally infected tissue, for the T cells to eliminate every virally infected cell, you really need to get a swarm of T cells in there to really clean out an area. you cleaned out that area, then another area may form and so forth until you clean out the whole thing. You can't do partial jobs. So positive feedback loop is probably required for that. So I think that's kind of the goal of the immune hub. The second hub we found with the TCF7 with the progenitor cells, that's probably is a more protected one, and that's what the one we're trying to understand right now, probably is similar to the TLS in some sense, because you have essentially the progenitor cells which aren't directly interacting with the malignant cells, and the dendritic cell becomes like in the lymph node. The dendritic cell usually moves from, let's say, the tumor or from a tissue to the lymph node, stimulates these cells to activate them, then they leave and they go and act and do their job. These small hubs that we found with the TCF7, we believe are playing the role of kind of a mini lymph node, and that is the belief of what the TLS is doing to some level. So it may be very similar, but there may be more of these, and they really haven't been appreciated as much as the TLSs, which are big and very easy to see and identify.
Great. So Ashok Khatri is asking, well, great talk. Are there any CXCL12 hubs?
I don't think, I wouldn't be surprised if there were, but we haven't focused on them yet. So I don't know the answer, but we can certainly check the data. Yeah.
So Nicolette Fonseca is asking whether this CXCL13 program is a specific, so if it is a specific for MMRD tumors or potentially a commonality in other immunogenic tumors, such as BRCA deficient tumors.
Yeah, this is a great question. No, so we find the CXCL13 program is present in any immunogenic tumor. It's just that we discovered most definitively in the MMRD versus MMRP. It was just a very big differential and easy to see the difference. But the lung cancer studies that I showed were not MMRD, those non-small cell lung cancer patients, and the breast cancer patients as well. So I think that they're going to be present. And there was a paper for Ziming Zhang a few months ago suggesting that CXCL13 may be literally the best marker for tumor reactive cells in any condition. So I do think that that's likely to be a general rule. And I think that these hubs are going to be forming anywhere there's a positive feedback loop that is going to be productive, so immunogenic tumors. I think these hubs are going to be present in all of those.
Cool. Pinji Lee is asking, are there any CXCL13 positive CD8 T cells in the tumor draining lymph nodes? And if so, do they originally come from the lymph node?
Yeah, that's a good question. We have not looked at that in the human system, and we haven't studied them in the mouse because I don't think that the same markers will hold there necessarily. But in the human system, I don't know. There are several groups right now doing lymph node studies versus tumor, and I do believe some of them have seen CXCL13, but I have to go back and check. There are a couple of papers recently that have come out. And I think there's also a question in the chat.
There's a question in the chat. Oh, yes. Do you see differences in immune responses in tumor cells depending on the characteristic of the tumor cells?
Yeah, that's a good question. We have not looked in detail at the genotype of the malignant cells and asked how does that affect ourselves in this study. But certainly there have been studies showing that KEAP1, for example, mutations will change the responses in immunogenicity and will change kind of local innate immune responses in the tumor. So I do think that there's going to be a whole set of oncogenes. There have been a few studies over the years that have shown that can affect the innate immune response of the malignant cell itself. And I think those are going to be very important for outcome that's been seen for some of these oncogenes. And, I think you can ask the question, how many oncogenes and tumor suppressor genes are affecting these responses? And I would guess that many at the end, if we kind of pull them all together. P53 does, I think. MYC does, many others, and so the RAS and BRAF. So I think that it's a good question, and I think that more deep understanding will come in the next couple of years on this question.
Cool. I guess I can ask. I know there's another one. So Jonathan Fradin is asking, thanks for the very interesting presentation. Do you have any insight into whether the biomarker program for PD-L1 therapeutic response could be detected in a CGP panel, solid or liquid?
I'm not sure what a CGP panel is.
Yeah, me neither.
But I think that if the question is, could it be detected in liquid? I think that, yeah, I think some of these markers, chemokines, have been found in ELISAs and proteomic analysis of the blood. So I think that can be possible. I don't know if it would be possible to do at the RNA level, but there are some studies in the last several months I've seen suggesting that there may be also RNA that may be detected in liquid as well. It might be possible to detect some of these markers. So I think overall there's definitely a possibility of creating panels both for the tumor itself as well as the blood for these markers.
Okay, now I'm gonna step in and ask I have like a lot of questions, but I'm gonna try not to go too long. So it's fast. So these hubs, it's fascinating, and I was wondering. I have like so many questions. I don't know which one to ask first. Let me start with the dendritic cells. So I guess those are cross-presenting dendritic cells, the ones that you see close to the CD8 T cells. Is that right?
That's right. Yeah, they're LAMP3 positive, CCL19 positive.
And then are you considering any? So I understand the feedback loop, but are you also taking or thinking of, the metabolic milieu and other, other things that could be also affecting where these specific hubs show up? Like, for example, do they show up close to the vasculature or far away or anything like that?
Yeah. So, I mean, again, the mouse study that was done showed that they were perivascular, these CXCL9, 10, 11 hubs in the mouse. And so, we're certainly looking at that now. We hadn't looked at that in the first couple of studies. So I think there's a reasonable chance they could end up being there. And so, yeah, I think the metabolic needs of creating these hubs is probably very high. These are pretty high energy situations where the T cells are moving quickly between one cell and another and, producing effectors to destroy it. So I think it's a very good point, and probably one that's important for kind of interacting with angiogenesis potentially, as well as with the energy needs that are required. So, but we have not specifically looked at the hubs relative to the metabolic states around them. But I think it's a great question.
I will be then waiting for the next paper where you show that the metabolic milieu is also influencing these hubs. There's another Nir and asks, did you account for possible tumor-centric determinants that can shape distinct immune states and responses?
Yeah, I think it's that's funny. That's the same question I think as the previous one.
Yes, it's kind of similar, yes.
Yeah, tumor-centric determinants. I mean, other than the oncogenes and tumor suppressors, which I think are profoundly affecting it, the other one would be the tumor mutation burden. So, I mean, that's really not something I talked about today, but the MMRD have a higher neoantigen count. And of course, that in itself is important because it leads to more antigens. So I think, that's tumor intrinsic as well, in that the more neoantigens you have, the more likely you are to have T cell response, but this could also be affected by expression of lineage antigens. There could be many different antigens that affect it. So I think antigens on the one hand, and then the circuitry of that malignant cell, which could be related to its lineage, or it could be related to the oncogenes and tumor suppressor cells, genes and mutations that develop. So think there's a huge number of determinants on the tumor side. So I think it's a key point.
I have to say that I would be curious if you have Ki-67 in your panels to see whether
Ki-67, I do, yeah.
If the tumor cells have Ki-67, where these hubs show up, or if they are, I mean, if there's like a competition as tumor cells grow for nutrients, or because proliferation is not evenly distributed within the tumor. Like tumor cells don't proliferate evenly. You have like hubs, I guess I can say, of proliferation. Yeah. So whether these immune hubs correlate or anti-correlate with these different states of tumor cells.
Yeah, that's a good question. I don't remember seeing a correlation between the proliferation of the tumor there and not. So good question. Can look at that.
Awesome. So if there are no more questions, I don't see anything else.
I think there was one more on the Q&A at the end.
Another
there's
one?
A few more. Yeah, there's a few more.
Oh, really? They don't show up.
There's one from, are the immune populations changed pre and post?
Oh, yes.
ICI treatment. Yeah, I mean, that's a good question. Do they change? I mean, acutely, I'm sure they change. We haven't looked at very short time points after anti-PD-1 in humans, but certainly in general, you're going to get a burst of proliferation and more exhausted tumor-reactive T cells in a tumor. But then what we find when we look in patients is that they kind of settle down to their pre-therapy numbers actually after a bit of time. So I think acutely there's a difference, and then it goes back to where it was somehow in some homeostatic system that brings it back, which we don't understand why that happens.
Cool. So now I do believe there are no more questions. This was really, really, really interesting. Thank you so much. This was an amazing presentation.
Thank you.
Oh, there's one more message. Oh, great talk. No, this was really, really interesting. And so, yeah, so interesting, for a better word, to see that in an unbiased way, you can identify something, right? Like at the pathology level, that it's, I guess, it was not visible to pathologists, and now you made it visible and something that can be accounted for. This
exactly, it's seeing things that were previously kind of invisible.
Yes.
I think is the key, and it really took diving into the gene programs to observe it ultimately. So yeah, that was the key point, and it allows us to kind of listen in on the dialogue between cells, basically. So.
Yeah. I like that, listening to the dialogue to find this. Yes, this was really cool. Thank you so much.
Great. Thank you so much.
Thank you,
everyone.
Bye.
Bye.